Thank you. I think I'm back on here. Thank you, Ellen. That's great. I think the Lab Partners program is terrific, and I want to thank Len and Ellen for their participation, and I want to encourage everyone else to join in. So, so it is now my great pleasure. So now I get to switch from being director to being part of the third of the day's dog and pony shows, right? But this one is going to be much more like a dog and pony show, I assure you, because so now I get the chance to introduce my good friend and colleague and collaborator of 22 plus years, Sherman Silber. And actually, Sherman is going to have, if it may become clear across the course of this interaction that we have a somewhat irreverent friendship. Okay, so we'll try to maintain some boundaries of civility on this, but if it breaks out of that, well, you'll have been warned in advance. So Sherman is going to have a proper presentation with some slides and things. I have decided that I'm going to provide color commentary on Sherman's presentation. So I may interrupt Sherman, correct things, or who knows. But in any case, and so the only formal visual that I have prepared is this timeline up here. So, so I don't know, I know the sight lines are a little tricky, but the timeline ends in 2014. But it has its beginning in 1992. And just to sort of add my... It doesn't end in 2013. It doesn't, it's currently, it's up to, it's only, we have not updated it beyond 2014. Exactly. Thank you, Sherman, thank you. You see how this is going to go. So, so I started here at Whitehead, as I explained this morning, I started here at Whitehead in 1984. And it was only when we were preparing for this, I began to realize I was here at Whitehead for 8 years. And then in 1992, two big things happened with respect to this dog and pony show. My laboratory published, in a pair of papers in Science, we published the first map of a human chromosome, the first comprehensive map of human chromosome. It was a map of the Y chromosome. And it turned out that a month or two after we published that, I went to, I think it was the American Society of Reproductive Medicine. I think it actually had a different name at that time. It was called the AFS. It was called the American Fertility Society. Went to the annual meeting, and I gave a talk about this work on the Y chromosome. And this person, Sherman Silber, came up and talked to me afterwards and said, well, I'll get into that. I'll get into that in a bit. But in any case, so in 1992, we published this first map of human Y chromosome, and Sherman and I met. And what we're going to do is tell you a little bit about what's transpired over the last 22 years. So I have to say a couple of things about Sherman in introducing him. First, Sherman is a consummate clinician. I would say that Sherman is the best infertility doc in the world, bar none. And I've met a lot of them, and traveled widely in these circles. And I think Sherman is known as the absolute innovator in this space. And he is brutally honest in his assessment of whether various treatments for infertility work or do not work. And as Sherman may tell you, there is a lot of nonsense in this field. There is a great deal of nonsense in this field. And one of the things that has most impressed me, and it began to impress me right away, is Sherman absolutely cuts like a knife through that nonsense. Now, it's not just that he's evaluating what others are doing, but Sherman has himself been an incredible pioneer of new surgical and microsurgical techniques. Sherman came out of a urology training, right? Actually, most of the people who work in the assisted reproduction world come out of OBGYN. Sherman actually came out of urology, out of a more strictly surgical and actually male surgical perspective. But Sherman has made, and it'll probably come up in your presentation or along the way, the number of specific advances, surgical and microsurgical advances that Sherman has made over the years is incredible. So consummate clinician, respected around the world as a foremost authority. He has also been he's been very active in spreading the word to the public about what all of these questions are about fertility and infertility. Sherman has written some very popular and very accessible books about how to get pregnant.
How to get pregnant.
How to get pregnant.
It's called a masterpiece.
A masterpiece, right. And Sherman has appeared on the Today Show, I think with Oprah, and on and on and on. So he's been very successful both in sort of getting the word out about what this means, but he is in fact a frontline developer of these technologies. But then the piece that really has made our relationship is, I go back to that first meeting. So I had just presented to the American Fertility Society, now the American Society of Reproductive Medicine, some talk about the cloning of the Y chromosome. And frankly, I was thinking, what am I doing at this meeting? What do I have to do with fertility and all this? But I had in the back of my mind, maybe we should be doing some more work on male infertility. But I thought, the trick is, this meeting is full of hundreds, if not thousands, of clinicians. And I know I like to pick my collaborators really, really carefully. I have no chance in this crowded ballroom of figuring out who I should work with. But anyway, this guy comes up, starts asking me, he says, is it all right if I call you David? David, do you know that chimpanzees have huge testicles? And do you know that gorillas have tiny, tiny testicles? I think that spermatogenesis must be genetic. And he said, I've really been interested in these questions about why the gorilla has the tiny testes and the chimp has the huge testes. I've been interested in this for so long. And I said, this is the guy. This is the guy. Sherman.
Well, thank you very much, David. It's been really a great joy for me to be able to work with David because I have clinical. Is that on now? Okay, and you can pick up the sound on the machine also. So we have been really, really close friends ever since 1992, and my first slide will give you an idea of just how close we've been. You really can't separate the X and the Y from each other. They each have different functions, And Dave has about 1,000 genes working for him, and I think I have 60 genes working for me. But this close collaboration began, David didn't mention when I told him, is that for years, the urologists and gynecologists have been treating male infertility a really stupid way, giving hormones or operations like varicocelectomy and all kinds of nutritional supplements. I can't tell you all the stupid, billions of dollars of stupid treatment which in control studies we found never had any impact on sperm count whatsoever. And so the whole thing was very unscientific and obviously very costly to the public. And I said, all that I can get from this, since it seems like every man's sperm count is pretty much constant and immutable, is there's got to be genetic control. And now here you've mapped the Y chromosome. Now we all know he picked the Y because it was the shortest, so he thought it'd be the easiest chromosome to map. It turns out it was the most difficult chromosome to map. But so I suggested to him, I have very loyal patients. They've been waiting around for 20 years, literally hundreds and hundreds of them with zero sperm. Let's pick a perfect phenotype that is really pure, where we really have a chance at this, and let's map their Y chromosome and see what we come up with. And 3 years later, we really changed the world of treating male infertility. And I'll get into that into a minute. Now, my next slide just shows how confusing, a time that really brilliant molecular biologists have really understanding the difference between men and women. They still don't get it. And in fact, they often ask the question, maybe it's the Y chromosome, why men and women are different. And I'll go through my funny slides first, and then I'll get into serious stuff. But so first a little fun and speculation, and then to substance. Now, we know that men and women really are not different. If we look at what is going to become the testicle in the fetal male and what's going to become the ovary in the fetal female, seminiferous tubules of the testicle are just ovarian follicles. So we know that men and women really aren't very different. Something happens around 6 to 8 weeks of fetal life in which the male has a testicle and the female has an ovary. Now, there's much more interesting things going on, but I'm just pointing out that men and women really are not that different. And in fact, this is a typical male that I've operated on. That's the testicle below. Up above is his epididymis, and you see that I'm picking up what is his uterus, upper vagina, and fallopian tubes. In other words, there's a structure called the appendix testes inside the scrotum of every single male we operate on, and that is the embryologic vestige that just shrunk away to nothing of his Mullerian duct, which is the uterus, upper vagina, and fallopian tubes. So I always tell men that inside in case they're not sensitive males, inside the scrotum of every man is a little woman. Now, we do seem different, though we are not. Completely different. And of course, there are these gender-specific views of how men function, but we all know that these gender-specific views are completely incorrect, And, we think of sex as being this sexy thing we see on television or in the movies, but really, here you can see these fish underwater actually having sexual intercourse, at least the way fish have sexual intercourse. In their absolute romantic excitement, they're doing whatever they can to provide for the next generation, and they are enjoying and loving every second of this last bit of their life. So even in fish, why is sex so universal? What's so important about sex? It creates all these problems, headaches in the female. And then, of course, these guys can't really be serious. And when giraffes try to have sex, I don't have pictures of it, if a giraffe falls down, he can't get up again and he dies. So he's risking his life trying to mount the female, because if he loses his balance and falls down, he'll never get up again. So why all this trouble over sex, Now, the other question that comes up always, why are the great apes like us or not like us? So here you see this enormous, huge gorilla, powerful but peaceful animal, non-promiscuous, very faithful, married to life. But he, as I told David, has very tiny testicles. He has a very threatened fertility. Well, why? Well, because he's so non-promiscuous that if his female partner is ever going to get pregnant, it's only going to be with his sperm. So he doesn't need very many sperm. Sooner or later, one of his little sperm is going to fertilize that one female. So his non-promiscuous, peaceful behavior as a gorilla is leading to his extinction. But then what about these chimpanzees? We're all so very proud that we're like chimpanzees. Why are we excited that we're like chimpanzees? I mean, I just asked David, and he said we really are genetically like them, but they are small, they are warlike, they are vicious, they're promiscuous, family troop life, and any female that goes into heat gets mounted by that male, and as a result, all these little 100-pound chimpanzees you see at the zoo, look a little closely next time. Just look closely, and you'll see they're dragging around on the floor of their cage. They're dragging around these grapefruit-sized testicles because, if any sperm is going to make it, it's most likely going to be, the sperm from the male with the highest sperm count. So these chimpanzees are really not, I don't know any male that has a testes like this, but every now and then I see one in the operating room theater, and I know pretty well what kind of life his culture led to. But anyway, so there are great apes that seem more like us, although David tells me their genes are a little more distant, but the orangutans ponder, and I frankly like them more for relatives than any of the chimpanzees that I really don't want to be caught dead with. Now, humans are different, and as a medical doctor, my goal is, with the collaboration of science, is to help humans. You'll notice that these are the only animals, humans, who actually have sex facing each other. Now, that's extremely important, the only animals, because the human is a peculiar animal in which the success of its offspring depends upon a family relationship in which there will be teaching going on for some time. And so it's important that for us, sex is not just chemical attraction and a giraffe trying to jump up on the back of another giraffe and risking his life doing so because the chemicals are stirring him to have sex. I mean, there are other things attracting us, and those other things result in a family life. Now, so let's go to the beginning. I want to just entertain you with how many questions there are to ask about the difference between men and women and the difference between all these different animals we're studying to try to understand human reproduction and realize men are not very different from women. Animals are not very different from humans, and studying animals led to all of our breakthroughs in human disease and infertility. So in 1992, I want to emphasize clinical translation of what we're doing, because the work here is very, very scientific and very, very, seems difficult to understand often, but there's always a vision in mind. And that vision should usually be far off. If it's too close, you'll just be doing pedestrian research. But if there's a vision far off of the bigger picture, sooner or later it winds up to have a clinical value that changes the life of human beings. And I think that's what we've been doing here at Whitehead. So in 1992, all the doctors thought sperm count could be manipulated hormonally. And this never worked in any randomized controlled trials, even though it was used everywhere, and billions of dollars were spent on it, and people have wasted so much money, and nothing ever really worked. In fact, some of the things they did, like putting men on HCG or doing varicocelectomies, would lower their low sperm count to 0. So after the initial Page Silber study, everyone realized that sperm counts were genetically controlled, and this revolutionized thinking and treatment. And this occurred because the Y chromosome was the first one to be studied. Why the Y chromosome? Well, just to give you a little lesson, the X chromosomes in the female are like ordinary autosomes, about the size of chromosome 7 or 8. They're pretty big. And as they evolve over the course of 300 to 150 million years ago, so that because they don't recombine, because you have a sex-determining Y chromosome, the Y tends to diminish in size because recombination is where repair occurs. And so a universal event, and I won't say in exactly in all animals because David has some new information in non-primates, but at least in primates, there's the evolution always of a sex-determining chromosome, and it evolves over and over and over again in all animals on the planet. There has to be some benefit to the evolution of a sex-determining chromosome, and atrophy occurs due to the failure to recombine with his partner. If the sex-determining Y chromosome doesn't recombine, then its DNA damage during the lifetime of the animal is not corrected. So the Whitehead was the first to map an entire human chromosome, which was an amazing feat. And one might say, oh, well, great, you mapped a Y chromosome. And what difference will that have on our human life and patient care? Well, like I say, you don't often see far enough unless you're really standing up high, and David is standing up high. So seemingly, the best first target for mapping a chromosome was actually this Y chromosome. But that's what led to our initial working together, because with 23 continuous years of research, it looked like that was a really good place to start looking for what I was convinced was the genetic cause of male infertility. So we convinced the geneticists that male infertility is not a phenotype, because if you had just mixed up a lot of male infertility, you would not have gotten a result that was meaningful. But non-obstructive azoospermia, that is the severe degree of infertility, is a reasonable phenotype to study. No control treatment for male infertility works, must be genetic. Y is the most likely locus, so all we had to do was provide the patients. And this was the map of the first study that came out in 1995 and has just got a gigantic impact factor. And it was the first demonstration of the genetic control of sperm production. And before Whitehead, no one thought that male infertility was genetic, and billions of dollars was wasted on foolish treatment. Before Whitehead, everyone thought that the rare testes sperm could not fertilize or result in a healthy baby. But after our Whitehead study, everyone knew that men with absurdly low sperm counts, essentially nothing, could have normal fertilization pregnancy, and that was with the invention of ICSI. So this is where we can pick up a single sperm from the testes if necessary and inject it into the egg and get normal pregnancy rates. So before Whitehead, it wasn't known that azoospermic men actually have sperm, but not very many sperm. Azoospermic men have sperm, but tiny quantities, but enough for ICSI, and not known before Whitehead. This was our first graph of it, and we really didn't make any sense of it. We published it in 1977, and you realize, if you look at it closely, but we didn't look at it closely when we published it, you could correlate the sperm found in the testis to the sperm count in the ejaculate, but we never realized how many cases where there was no sperm in the ejaculate. If you look on the left-hand side, there was still this incredibly tiny amount of sperm in the testes. So all we had to do is microsurgically explore through the testes, and I provided that kind of technical skills, but David and the group here provided the science behind it. So if there is before Whitehead, everybody thought that there was this huge DAS deletion, which we discovered in 1995 on the Y causing azoospermia. Why should there be any sperm at all in the testes? And no one knew why, except that there was this original drawing of this crazy appearing initially structure of the Y chromosome with all these repeats called amplicons, DNA repeats that were really unprecedented anywhere else in the human genome, and really defied sequencing in all the rest of the Human Genome Project. But, and these were recombination targets where areas would drop out and we'd be missing huge amounts of genetic material that would cause azoospermia. But if you look at the next graph, what David found was that there were homologs of the DAS gene in the autosomes, and these homologs were able to act as backup. So you wouldn't have a very high sperm count, but if you look carefully, you'd find sperm in these men. Not very many, but enough to get their wives pregnant. So you could have degrees of azoospermia depending upon how many backup homologs there were to help in spermatogenesis, which was really a multi-dosage phenomenon. So on a testis biopsy, you could see Sertoli cell only, mostly, but if you look very carefully, you'd find areas where there were sperm, and you could pick up this sperm with a little micropipette, and then you could inject those sperm into the egg. Now, this looks kind of scary to most men. It's really painless.
That's right. It's time to cross your legs. That's right.
You can tell by the fact that men are willing to put up with this who runs most families. And no, but this is a picture of how it's tolerated by men.
Just as with vasectomy,
Hopefully
as
you can hear
performed
the
in the
sound.
doctor's office, the cord is grasped
I don't
between
hear
the
the
thumb and the
sound.
forefinger, and
It's low.
0.5%
Let's see if
marking
we
is
can get that up.
injected.
I don't know if we can get this sound up.
Well, at any rate, yeah, okay. So I'll go to the next one, and you'll see that he can, it's
The
really not
patient
that
is able
Here
to
we go.
actually view on the video screen the microsurgical anatomy of his own obstructed epididymis. He has congenital absence of the vas, as
So it
well as
doesn't,
congenital
it's not as
absence of the
painful as it
distal
looks, because we do it under local
We're demonstrating
anesthesia.
to him that if we were to try to retrieve sperm from the distal epididymis, they would be old, senescent sperm, or even just sperm parts that had decayed over a period of
And
time.
now I'll go on to what it looks, this is after surgery.
It's a low price to pay.
He's hopping off the table,
Okay.
You notice he's standing up straight.
walking out, great hat.
Is this before or after the surgery?
after the
This is
surgery.
after the surgery, right.
For all the future procedures, you won't need it anymore.
So it used to hurt a lot afterwards, but well, I don't know where this Callahan lady is, but the Department of Anesthesia, you've got to pay attention to everybody in the world from Cork, Ireland. I mean, whoever heard of Cork, Ireland? But they've got a fantastic medical school and a great department of anesthesiology there, and they developed this long-acting anesthetic. So now these men don't hurt afterwards because it lasts for 3 or 4 days, and by then it's healed. I mean, they used to kind of walk around, you know, a little sheepishly for a few days, but we no longer have to deal with that. That's progress. So, so often you can see in a testis that's got maturation rest or making very little sperm, you can see a tubule with completely normal sperm, and I believe it's because of these, when you have DAS deletions, it's because of these homologs on the autosomes that take over. Now, before Whitehead, no one understood the complex nature of the XY marriage, the evolution of spermatogenesis genes of all species gravitating to and concentrating on the Y was really unheard of before, and the concentration of spermatogenesis genes on the X was just a fairy tale, but it turns out it was true. So does the understanding of the evolution of the X and the Y chromosome, which sounds like it's very scientific, help translate for the clinical doctor like me into treatments, often dramatic for patients? Well, yeah, I mean, it absolutely does. Let me give you one example of a rather obscure study. This is an azoospermic patient, and we were able to get sperm from his testicle. And of course, that was before the cork discovery, so he walked around a little bit funny for a while. But nonetheless, he was able to have a baby. And we looked at it, and we found that the father, the patient had AZFC deletion. The boy had an AZFC deletion, so no big deal. He'll just have to go through what his father went through. And you can see that the father and all paternal uncles and brothers are obviously fertile. And you can see in the father's blood, no AZFC deletion, the father of the infertile man. But his sperm had no AZFC deletion either, because, I mean, we can't look at all the sperm. If we looked at 2,000 sperm, it would be a different story. So, therefore, what we really discovered is that this is de novo. What it means that in every one of you normal fertile guys, probably 1 in every 2,000 of your sperm are going to have some sort of deletion because of the structure of the Y chromosome. And if that's the lucky sperm that fertilizes your wife's egg, then you won't know it, but your son will grow up to be azoospermic. But thank goodness now we have a treatment for that. So about 1 in every 2,000 sperm in an otherwise fertile man will carry infertility. So it doesn't seem like it's inherited or genetic, but it obviously is a de novo genetic deletion. Before Whitehead, there was nothing you could tell this patient, and our ignorance would cause anger and misery, because he said, there's no history of infertility in my family, and he'd be angry. But now after Whitehead, we can give him an intelligent answer, and we don't deal with that anger. So now the question, how did the X and the Y evolve? Can basic understanding of science prevent foolish and vastly wasteful treatment of the patient? Again, I'm focusing on translation. And this may be too complicated a slide, but you probably all know from David in the past that the appearance of this male sex-determining gene maybe 300 million years ago resulted in degeneration, and that degeneration of what was a normal autosomal pair turned into the Y chromosome, which is me, and the X chromosome, which is David. And that continued evolution is very interesting, because when you lose all these genes, how is the male going to survive? Well, and then that came out of this lab, because no one understood it, and it was very confusing. It didn't make sense, but that came out of this lab, that the X chromosome hyperactivates at the same time. So that saves the male, because he only has one copy. And in the female, X inactivation occurs so that half of them aren't really expressing. So it was an ingenious mechanism that developed over time, so that despite the fact that, for example, men only have one intelligence gene for every one of women's 2 intelligence genes, despite that fact, we seem to have equal intelligence. And that's true really of all of our function. And so that explains some real confusion. So why does this Y chromosome not just completely disappear? And the reason it doesn't completely disappear with evolutionary pressure, I will say, is that male fertility genes are located on these palindromes, and they survive by masturbation. I'll explain this better. When you have multiple copies of genes that are controlling spermatogenesis, multiple copies, and they're in inverted relationships like palindromes. So instead of the Y can't recombine with the X, so it recombines with itself. So autosomes repair themselves by recombining with each other during meiosis, and the Y tries to repair itself by recombining with itself, but it can't. So autosomes have sex with each other, and the Y just has to masturbate. But either way, you're able to keep it going. So before Whitehead, there was no idea what the Y chromosome looked like and how significant its role is in human disease. It's impossible for any other lab to actually sequence the Y. So that would seem like a very basic science, obscure thing to do, except it's had its enormous translational effects that many people couldn't have predicted. So we actually have really, really good sequence that's reliable, and we know all the amplicons and spermatogenous genes on these areas of multiple repeat sequences. So the Y amplicons defy routine sequencing, and 12% of the X is similar and also defies routine sequencing. Now, I don't know if David agrees with me, but I have this just gut feeling that that 12% of the X, which nobody has been able to sequence yet, and we haven't sequenced yet because there's a million other things to do, could very well be the dark side of the moon, and there could be a lot sitting there. So what about the X chromosome? Like the Y, the X chromosome is present as a single copy in the male. Therefore, mutations on the X that are favorable to spermatogenesis will readily be expressed in the male offspring. The X spends 1/3 of its evolutionary life in the testes. In other words, it's not always in an ovary, where it cannot recombine because it's with a Y. And so it only makes sense that the X should have a lot of palindromes and should have some kind of a compensatory mechanism like the Y had to develop to survive. And so the human genome really has not been completely sequenced, and we may be the only ones that really know that. So I don't know, I think of that as the dark side of the X, that are very important. Now, there's two important new human scientific projects we're working on, and then I'll try to end it so I don't go over my time. But oocyte arrest in the fetus and oocyte recruitment in the adult is crucial for what I do every day, IVF. And 6% of babies in the world, remember, are IVF babies. And our biggest problem is the vast majority of IVF embryos obviously are not viable and don't take. So IVF is costing a fortune for couples who are crying because it's not working for them. Not that it isn't successful, but in the majority of cases, it really isn't. The majority of human embryos from IVF really aren't working right. And they look absolutely beautiful, and they all ought to be working, and what is really the problem? So that's an important question. And furthermore, none of us understand when we stimulate to get eggs for IVF, or even when you have normal stimulation, like with your ovulation every month, what is recruiting these primordial follicles? You know you have about 400,000 follicles as a teenager. 1,000 die every month that are recruited, and one of those recruited follicles actually ovulates. What is the mechanism for recruitment of those resting or arrested primordial follicles? What's happening? There is absolutely no hormonal stimulation. No one has figured out, no matter how hard they try, has figured out what is it that recruits these 1,000 follicles a month. No one understands it. And yet they all die off, and that's why women become infertile as they get older and at age 50 go through menopause, because no one really understands what's going on. Why are these eggs just disappearing on us, 1,000 a month? What's really happening? We don't understand that, and now we do, because of embryology work that was begun at Whitehead in 2006. Also, we have the poised genes issue. I am interested in what it is when we see a beautiful embryo, why these embryos really don't develop normally and become babies. Something really pretty amazing. I'll go into that in a minute. First, the poised gene project that Bibi Lesh is working on, and that David, I think, is one of his favorites. Sperm and eggs are high. It's very simple. At first, they had explained it to me 100 times before I understood it, but now I get it. Sperm and eggs are highly differentiated cells which are very, very limited in expression to very specific functions. I mean, a sperm is a funny cell that has one job to do, and it has one differentiated function. And an egg may look like a great big thing, but it's very, 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 very complicated and very, very specialized. So how do they suddenly become totipotential cell when they fertilize that can develop into a rapidly developing embryo the second they fertilize? And it's got to be these genes that are not just turned off in the usual way, like you hear about turned off genes. They've got to be turned off in such a delicate way that they can go in either direction extraordinarily rapidly. So David calls that poised genes, and I guess it's because it's methylation of the histones rather than DNA itself. So how are they going to really able to prove that? Well, here's where I get into it. Not only am I interested in why we can't get these women pregnant, but in order to really demonstrate this happens in the human, they need viable human healthy tissue. Well, that's where I come in, because how are you gonna go down the street and ask men to submit to a study where we're gonna do a testicle biopsy on them so we can figure out why embryos develop right or don't develop right? Well, I mean, I have this captive group of vasectomy reversal patients that come in, and they've been signing this consent form for the last 30 years, 37 years actually, saying that we will agree to do this, and it's reviewed all the time by RRB. They will agree to do this if we can use a biopsy from their testes for research. I didn't know what it was going to be, but you're not going to be looking at human testes like this, twice a day and not find some use for it. So therefore, we become probably the only source available for David and Bibi to figure out whether or not these genes are turned off during gametogenesis. So a fantastic collaboration I never thought was going to develop, but our availability of human material, which is very, very ethical, can be tremendous for them. Next thing is, you've all heard about my ovary transplant work. Well, something very interesting happens with this that's helped us understand why these eggs die on you women and why you have to go through menopause and become less fertile when you're older. Before Whitehead, there was no understanding of the embryology of fetal egg arrest. I'll summarize briefly. Oogonia enter meiosis at 6 to 8 weeks under the adrenal bombardment with retinoic acid in the fetus, which upregulation of STRA8, and that makes for meiosis. So once these cells begin meiosis, what's supposed to stop them? I mean, they're supposed to keep on developing, right? Something has to stop them, or else the baby girls will be born without any eggs, you know? Well, okay, let's look at the male first. So in the male, there is no problem. The outer cortex of the male is the same as the outer cortex of the female. The outer cortex of the male testes is designed to protect us when our wife is so angry she kicks us in the testicles. But for females, you don't need such a stiff cortex for that, but you need it for something else. Sertoli cells protect the primordial germ cells in the male from that retinoic acid. So we have stem cells and we make sperm until we're 120 or so. But ovarian follicles, they're going to all disappear. They must invade the tunica albuginea to arrest their continuing meiosis. This testis tunic thus becomes the ovarian cortex. So after Whitehead, what we could know is the development of ovarian freezing and transplantation is a tool for understanding ovarian recruitment and explains the mystery of adult resting follicle recruitment. The transplanted ovarian cortex encounters a sudden release of lifelong pressure of control of primordial follicle arrest, and we can see it with my last slide biochemically. As the FSH, which is red, comes down to normal, and most of these patients get pregnant spontaneously, but they all develop normal ovulation and normal menstruation. And this is from frozen, this is actually a multiple sclerosis patient who had her ovary frozen several years ago, had it transplanted back. But you see there's this massive recruitment in blue, way more than normal, of AMH, indicating with pressure release that suddenly all of these follicles, which were released gradually like a teaspoon at a time, one egg at a time, suddenly are massively released. And so now we have an understanding through our ovary transplant work what ovary recruitment is all about. And one of the real tremendous applications of it is you can take a woman who's 35 with premature ovarian failure, and there's always what I would call, you can always find what I call a dead-looking egg somewhere there in the cortex, in the histology. If you look, they laughed at dead egg, but I mean, you see it and you wonder, what is this thing doing here? She's not ovulating. She hasn't menstruated for 10 years. She's in premature ovarian failure. What's this thing doing here? Well, now we know that we can cut off the cortex and chop it up, transplant it back, and she starts ovulating again. Because we've released these few eggs that are left in a young woman. It doesn't work in an old woman. But a few eggs that are left, she can start ovulating again. So that all came to us when we were starting to think about the fact that Whitehead discovered the nature of the initiation of meiosis in what in humans would be the 6 to 8 week old embryo. And that initiation of meiosis, which was very, very misunderstood before, created a new question. And every answer Whitehead comes up with creates a new question. And the question is, why don't those eggs disappear? Why aren't they gone by the time the fetus is born? So what we know now is that oocyte recruitment of resting follicles should be renamed release from arrest of fetal follicles. So that's my summary of, I think, a huge amount of work that's come out of only 20 years, you know, of working together. So David's younger than me, and I think I've got another 20 years left. So I hope to present a lot more material in 20 years. Thanks.
Sherman.
Okay. Any questions?
Yeah. So actually, Sherman,
Yeah.
why don't you
Okay.
sit over here,
Okay.
and because I'm just gonna pick up on a couple of the things that Sherman talked about and go back to this timeline.
Can I have a taller chair?
Yeah, you can have the right. So I want you to understand what a absolutely fantastic collaboration this has been over these 22, 23 years. And as you can tell already, that Sherman and I were made a logical pair because we have such virtually identical personalities. And we have such virtually identical backgrounds, and we could go on and on and on. Right? So we made quite an interesting pair over the years here. But so let me just pick up on some of the things Let me pick up on some of the themes that Sherman developed and just go back to this timeline and give you a sense of what this interplay has been like. And I should say that from the time that Sherman and I started collaborating in 1992, Sherman has come to visit us. So, Sherman has an absolutely crazy existence as a clinician, right? Tonight, he will leave after the program tonight to actually go down to New York City to do some cases tomorrow morning, to do some surgical cases tomorrow morning. He was in Hawaii a few days ago, but despite this sort of frenetic existence of the international clinician, Sherman is always calling me and saying, David, I want to come when am I going to spend this year, when am I going to spend that week with your lab?
Right.
And actually, Sherman loves to come spend a week or the part of a week each year talking to the trainees, the students, the postdocs in my lab.
That was my 60th birthday present.
That was your 60th birthday present. So over this, just to sort of review what sort of happened here, we said in 1992 we met, and it was sort of driven by this first mapping of a human chromosome. And by the way, just to give you another sense of what was the upshot of that, this first mapping of a human chromosome led to the growth of what would be called the Whitehead MIT Center for Genome Research, which eventually gave rise to the Broad Institute, right? So, but also I met Sherman that year, right? And began this collaboration. So as Sherman said, 3 years later, so we started collaborating almost right away.
Right.
Sherman, as Sherman explained, we had just developed this sort of basic scientist map of the Y chromosome. And maybe I'll make a sketch over here. So we had developed this basic scientist map of the Y chromosome. And I'm going to put over here this is the centromere. Over here on the short arm of the Y, I'm going to put SRY. That's the single gene on the Y chromosome that instructs the developing fetus to become anatomically a male, or in its absence, anatomically a female. But by 1993, working together, Sherman sent us blood samples, collected blood samples from many of his patients, men who had very poor sperm production. We prepared DNA, applied our recently constructed map of the human Y chromosome, and what we found, as Sherman has just described, we found that quite a few men with very poor sperm production were missing a chunk, had a deletion within the long arm of the Y chromosome. Okay? So and it turned out that just some years later, by 2001, we now had been pursuing not just the mapping, the rough mapping of the Y, but by now we were pursuing the sequencing of the Y chromosome. And what emerged in this region that was commonly deleted were actually palindromes, huge palindromes. And so I put up here on 2001 just the existence of palindromes. So you're looking puzzled there, John. So I got to tell you that on your Y chromosome, you have a 3 million letter sequence, which is 1.5 million letters in one direction and then flipped around and copied in the other direction. It's a palindrome. And there are 8 of these palindromes altogether scattered across the long arm of the Y chromosome. And it is, in fact, this palindromic structure of the Y chromosome which makes it prone to the generation of these deletions, which, as Sherman said, so it turns out it's actually 1 in 2,400 newborn boys. Oh, it's not, okay, I'm just to be precise.
Pardon me.
Yeah. 1 in 2,400 newborn boys is missing that particular part of the Y chromosome as the result of a fresh deletion on the Y chromosome. And then what Sherman did, so we then went back, having found that men with very poor sperm counts often were missing that part of the Y. The question, as Sherman said, was, okay, what is the clinical relevance of this? What can you do for those guys?
Yeah.
And Sherman already said there's no point in giving them and throwing hormones at them because they have a clearly genetic cause of this. But what Sherman then found was, remember when he had those surgical pictures where some of the men in the audience were crossing their legs? What Sherman found was that he could actually, in guys who had low sperm counts because they're missing this part of the Y, he found that in many cases he could actually do a testis biopsy and from that biopsy retrieve sufficient numbers of sperm to actually allow that man to become a biological father by single sperm injection, by the ICSI that he talked about. Okay, but at the same time, so we continued looking at other men who had who had problems with sperm production, and we found that there were some men who had an even bigger deletion on the Y chromosome. And what Sherman found is the guys with the bigger deletion, you can do the testis biopsy, but you will never find sperm.
Yeah.
So it turned out that was extremely useful information to have clinically. In other words, if you had this deletion, you're not going to make any sperm. There's no point in doing the testis biopsy. If you have this smaller deletion, what would you say is the probability? If you found a guy with if you have
small deletion, it's 86%.
86% of the men, you will find sperm. And so that was incredibly valuable information to put to use in the clinical setting. So that sort of gets us up. And I should say, when we found these deletions on the Y chromosome, this is now work that seriously began about 20 years ago. Our first publication in this area was in 1995.
Right.
It's an incredibly highly cited publication. And this remains to this day the most common known genetic cause of spermatogenic failure, the most common known genetic cause of male infertility. Right. And it's actually led to an explosion in the medical scientific literature studying Y chromosome defects. And in fact, so we have essentially provided the tools by which the entirety of the world now diagnoses men with low sperm counts looking at their Y chromosomes. In other words, and this is now standard clinical, this is now, we were actually talking about how you intervene here, but in fact now, if a man has a very low sperm count, it is now standard clinical practice to test them for points along the Y chromosome that we first mapped back in 1992 and sequenced 10 years later. So this is now standard clinical practice. And let's see, a few other points. By 2003, we got to the full sequence of the human Y chromosome, which allowed us to think much more broadly about anomalies on the Y chromosome in patients that Sherman was studying. And I'll leap out to even the present, where now we're thinking in 2014, we're thinking even about the role of the Y chromosome and the role of the X and the Y chromosomes in the enormous differences in males and females in disease beyond the reproductive tract. Okay? And in fact, we're collaborating on those studies. So we actually, all of this started in the context of sort of human reproduction and the sex chromosomes and thinking about the making of eggs and sperm. But you know, I could actually link it back to this morning. We were talking about autism, right? The 4-to-1. Why are males so much more commonly affected by autism spectrum disorders than females? So in fact, you know, basically in 2014, we are here with a new world of opportunities that include not just the reproductive tract, but way beyond it. So I could go on at great length. I think I'd rather just stop here, and I'm going to sit down with Sherman and see if you have any questions that you might have for us. Yeah. Okay, yeah.
Do
we have mics?
some way affect the gender of the fetus?
Where's the question coming
The
from?
question's up
Oh,
right
there, go
here.
ahead, say it again.
Can you have...
Does some way, some treatment affect the gender of the fetus?
Can you change the gender
What
of
would
the
otherwise
fetus?
be
Well, we never really thought about
5149
that.
or
It's
whatever?
easy enough with IVF to do a biopsy and determine if the fetus is a male or a female. But I don't believe in doing that because I'm a strong feminist, and if anybody asks me to do that, I tell them to go to a money-making clinic down the street, but not to me. And then it's funny, then these people stop and think about it, and most people know that men and women are equal human beings, and most of these patients that came and asked for sex selection come back to me and say, we don't care if it's a boy or a girl. I feel like I'm instructing them by turning them down. Now, in terms of making them into a male or a female, I don't have any interest in that.
Actually, let me see. We got the mics moving around. Okay.
Can I call you?
Well, absolutely. You can call me anything you want. You can call me Sherm.
Sherman.
You can call me Shorty.
My colleague Willem from Amsterdam and I spent yesterday at the Harvard Law School listening to a very discussion of the laws relating to surrogate parenting.
Mm-hmm.
And it seems like there are many problems in this area still, and it has not with the science at all. And the question I have for you, the example he was using is the fertilization of an egg, 12 of them embryos, and they were sold. And the question was, should they be able to be sold to women? And then this gentleman who created this wanted sort of to meet all these people and sort of have a family reunion, if you can believe this. And the question is, the law has a lot of difficulty with this, and I wondered if these areas of...
You always hear about these unethical people doing stupid things that all of us would have a difficult time with. And my son, who is about to graduate at Hebrew University Law School and has studied all these things, and they had an enormous study of gestational surrogacy, wrote a huge brief on it. So the problem with gestational surrogacy, there is no real problem in the United States except for New York. And the reason is that we don't go by the laws of legislators who don't know what they're talking about anyway. We go strictly by common law. And according to common law, the intended parent is the parent. So whether it's egg donation or whether it's gestational surrogacy, the opposite of it, it doesn't make any difference because common law and court law has always made it very, very clear and guaranteed that the intended parent is the parent.
So I've never been interested in doing uterine transplantation. That's only necessary in the majority of the world where there are laws against gestational surrogacy. Now, there are hundreds of thousands of women in the United States alone that really very professional agencies have contact with all the time that love nothing more than being pregnant and then giving away the baby. And so gestational surrogacy, some might consider it's abusive to women, but the women that do it absolutely do not feel it that way. I interviewed them all very, very, very carefully, and the parents get to know the gestational surrogates very well, the intended parents, and they've gone through a lot of psychological evaluation at our clinic, and I am sure that there are only a few cases you hear about that. Maybe if you go to a seminar somewhere where they want to create some excitement, where some very unprofessional doctor who maybe ought to be in jail has pulled off some stupid routine. But by and large, for a good, proper medical care, gestational surrogacy works very well, and we have at least, we have at least one or two or three every week. And there are several causes. One could be if a gay couple wants to have a child. Another, and I have nothing against that. I'm totally open to that. And the other could be if the woman has a uterus that really won't be able to handle a pregnancy, but her ovaries are okay and she's healthy otherwise. So there are many situations that come up where gestational surrogacy is a much better answer than what I think is the immoral option that got a lot of press from Sweden, just because they got this stupid law in Sweden where some stupid legislators who don't know a thing about motherhood and don't care about women really, and you know about Sweden's record on women if you've read enough about the girl with the tattoo. You'll know that Sweden has the worst record probably in the world on women's rights. And so they have these stupid lawmakers that make laws that are absolutely, that the laws themselves are abusive to women. So that in order to have a baby, a woman has to ask her mother or her sister to go through a big operation to remove her uterus, to transplant it to her, to put her on extraordinarily toxic immunosuppressive drugs, to risk an intra-abdominal catastrophe during the pregnancy which could kill her, and that's supposed to be morally okay, and gestational surrogacy isn't? No, I think what they're doing in Sweden and got all that publicity is morally shocking, really, but it's based on the fact that legislators in most countries around the world, who, like everywhere else, don't know anything, are passing laws based on their own concept of morality, which is usually anti-feminist. But at least in this country, with the exception of New York, where they had that Baby M controversy, in every state in the United States, the intended mother is the mother, and the gestational surrogate has tremendous protection against being abused.
You haven't seen situations where the mother, the surrogate, changes her mind. There have been a couple of those cases and doesn't want to give away the baby.
That only happens if you make the mistake that the egg donor is also gestational surrogate. Now, if you make that mistake, then she has a right to the baby and it becomes a legal mess. But if you do not make that mistake, let's say the intended mother doesn't have eggs left. So you get an egg donor, you get a gestational surrogate, and you use the intended mother's husband's sperm. Then the intended parents are totally protected, and the gestational surrogate has no right to the baby. And she knows she has no right to the baby, and it's talked about ahead of time, and the legal paperwork is obvious, and there really aren't any controversial cases around.
Thank you.
Okay.
John. Oh, well, why don't we go to Hillel first, and then we'll come over to John. You got the mic, yeah.
David, I think it's for you. You found a way with Sherman to have babies for couples, but is there a way to fix that deletion?
Oh, great question. So now that since we know exactly what the defect in the Y chromosome is, can we actually just go in and do chromosomal therapy? Can we repair that chromosome? No. Sounds simple, right? Like all I'd need to do is, I think, I just, erase this, right? And it looks, fill it in, right? Okay, I just did that. So that fixed it on the board, but it's really hard. If you have a piece of the human genome missing, which is what's going on here, it's really hard to put it back in. And also consider the fact that, I'd say in the larger realm of gene therapy, there has for a long time been the debate about so-called somatic gene therapy versus germline gene therapy. In other words, it might be one thing to add a gene to someone's, to some part of someone's body. It's quite another thing to add a gene to one's sperm and pass it on to the next generation, right? So when we're talking about fixing this, we'd say, which are the cells, the only cells in the body that actually need that entire Y chromosome are the cells that will themselves become sperm. Okay? So it's not as if it's the supporting cells of the testis that need that part of the Y. It's the cells that will themselves become sperm. So if you were to try to repair that, you would need to really be able to do it effectively and safely, because whatever comes out, you would be transmitting to sons. Okay, so that's it's a little bit of a ways off. But Sherman would say, look, not a problem. If it, especially, right, if it's the guy, this is the guy with the small deletion, right? Guess what? Those guys with the small deletion, Sherman can go in and find sperm in the testis biopsy, and by ICSI, by single sperm injection, become a biological father. Now, I want you to think this one through, right? If a guy has that deletion on the Y chromosome, which causes his sperm count to be greatly reduced, if you use those sperm in ICSI, what are the possible outcomes? This is a problem set now. I want you to tell me. Okay, so what are the possible outcomes? What kinds of sperm will this guy make? Half of his sperm will carry the X chromosome. What's the state of his X chromosome? It's a normal X chromosome. So basically, if you do ICSI, single sperm injection, on sperm retrieved from a guy with one of those smaller deletions, half the time the result will be a female and should have no problem related to the sex chromosomes. The other half of the time, the offspring should be it turns out, in the other case, will it be if you get this Y with the deletion, will it be a male or a female? So it's going to be a male because of SRY. This is the gene that makes that's going to make that baby an anatomic male. But it's still going to have this deletion. And so when that boy grows up, he's not going to make so many sperm. Now, Sherman hasn't quite yet figured out, or I don't know. Have you figured out yet when to tell these boys about their deletions?
Oh, I don't think you have to keep it a secret from them, but tell them that they'll have to go through what their parents went through.
Yeah. So you see, Sherman is creating a future market for himself in this way.
This is a growth industry.
That's right. Yeah, John.
So the success evident from your collaboration testifies to the power of putting clinicians and researchers together. And I think there are other examples in medical science. Judah Folkman coming together with basic researchers and making great advances in angiogenesis. So, does Whitehead have a program where you invite in clinicians to embed themselves with researchers for, say, not a week, but say for a semester?
So a great question, John. So do we have a program to embed physicians here? I would say we call it our graduate students and our postdocs. And so a large number of our, well, we were talking about, okay, let me link it back here. So Sherman was talking about our work, which I haven't said anything more about here, this poised genes.
Right.
Okay, we think there's a set of genes that are in a special poised state in the sperm and in the eggs. We think these genes are ready to go as soon as fertilization happens. We think these are the entrepreneurs that are ready to launch the whole developmental program. That work in my lab was done by an MD PhD, an MD PhD postdoctoral trainee who has traveled out to Sherman's lab to receive those samples from the vasectomy reversal biopsies. Okay, and so we have lots of, I would say, the trick is, it's just like when I was at that American Fertility Society meeting, and I was asking, with whom am I going to collaborate? The finding of collaborators is its own very intricate sort of mating ritual, right? I mean, you got to figure out who can I be simpatico with? And, as you can see, with Sherman and me, it's an obvious match. Right? No, but the thing is, we both really wanted to get to the bottom of this thing. And, Sherman was really unusual in that in the land of assisted reproduction, most people wanted to simply build their clinical practice.
It's a real serious problem. Clinical, by and large, IVF doctors are, by and large, just interested in making money and getting home by 4 o'clock, and they don't have an intense, they weren't the top of their medical school class usually, and they're just not that intense about understanding what is before them. What is before them is the most amazing opportunity to understand the most important aspect of human biology, and they don't take advantage of it.
But back to but John, so for me, so actually throughout my whole time at Whitehead, so actually once upon a time I went to medical school. So did David Sabatini, so did Rudolf Janisch. So actually our faculty is sprinkled with individuals who actually had some degree of medical training. As I said, we bring a lot of MDs or MD PhDs under this roof as trainees. And across my career, I've collaborated with dozens and dozens and dozens of physicians. And in fact, right now, we are at the beginnings. If we get the funding from NIH, Sherman and I are together at the beginnings of what we hope will be a very rich and robust collaboration, actually with some intramural scientists at NIH, at the Clinical Research Center at the NIH campus in Bethesda. We're collaborating with medical geneticists there to see if we can actually explore sex differences in disease. So, a bit of a long, Maybe you want to hand the mic down there to Herman and then back. Yeah.
Well, I would like to ask an irreverent question.
Oh, okay.
I've never done that before, of course.
Yeah.
An irreverent question?
An irreverent question.
Irreverent.
That's
the only kind that we allow.
Irreverent.
Right.
Okay, but I was just considering, as we went through this, with all the beautiful things you're doing about people who have fertility problems, with the population growth on this planet and the number of children that are born and need to be adopted and all other sorts of things, what is the rationale from an ethical point of view of trying to spend time and resources getting people to have babies when the planet has far too many and is projected to have even more,
Actually,
even
that's-
without any fertility problems.
Well, that sociologically and from an economics point of view, that statement, I'm going to be irreverent, is old-fashioned and incorrect. The modern view of actuarialists and economists is that one of the reasons, for example, we talked about yesterday, that Japan's economy is falling apart is because they don't have reproduction, they don't have immigration, women are not having children, and women can't even get jobs. So they have a decreasing workforce trying to support an aging, graying population. And that's why Japan, despite being the most efficient manufacturing and one of the most creative countries in the world, is in a severe depression. Now, one of the reasons that hasn't talked about a lot, that Spain and Italy and Greece have had a terrible economy, whereas Germany has held up very well, and in fact, Northern Europe has held up very well, is their declining population. So modern economists will tell you that a declining population is an indication you have an increasing number of old people who are not going to really develop the economy, and a decreasing number of young people who would develop the economy, but there's not enough of them to do it because they're really basically paying the pension of the older people who aren't contributing. So the world isn't Malthusian, actually, and the countries that are having a problem with overpopulation are undeveloped countries. I'll use the bad word undeveloped countries, in which there are massive public health efforts at contraception so that they can get a break on it. But the most common reason for seeing a doctor, say, in China is infertility, and the most common reason for seeing a doctor in India is infertility, because as a country is beginning to develop scientifically, they no longer need these 12 kids, 10 of whom are going to die, to run the farm, but what they want are those younger children that are going to grow their economy.
Let me actually, Herman, let me answer your question. Let me completely flip it around a little bit. So has anyone here heard of PCSK9? Does that mean something? Okay, so what is PCSK9? It is the hottest new target in the development of cholesterol-lowering drugs, and actually monoclonals and such. Actually, a number of the companies within our immediate neighborhood are madly they're in the race to develop the first blockbuster anti-PCSK9 drug. Now, how was PCSK9 identified as a potential target for lowering cholesterol? Turns out there were individuals who were mutant in PCSK9 and had incredibly low cholesterol levels. They had no problem with their cholesterol levels, but they illustrated by their own biology that if you wanted to lower cholesterol levels, you should target PCSK9, because they didn't have PCSK9 and they had very low cholesterol levels. So now, let me take your question and flip it around. Say, if you're concerned about problems of world overpopulation, perhaps we should be thinking about developing novel contraceptives. Where then would you look in the human genome for potential targets of novel contraceptives? I don't know if you see what I'm saying. So imagine then genes which, when defective, cause you to be unable to make eggs or sperm, or that interrupt the process. Those would be precisely where you want to intervene with novel contraceptives. So I will make the argument that this kind of research, which it actually works on both, it has potential application to both problems. That is, obviously there are parts of the world where overpopulation is a tremendous problem. As Sherman says, there are parts of the world where overpopulation is distinctly not the problem. But this sort of fundamental research into how do we make eggs and sperm can actually identify targets that work to both of these ends.
Well, my question was to a country.
Yeah.
What I read is the world population growth to hit 9 billion in a few decades.
Yeah.
Well, the best contraception and protection against world growth is a college education. It's been said for many, many years. And so if you can have development of a fair world in which it's not just Africans that are overpopulating the world or developed or undeveloped areas that are overpopulating the world, but you're really educating these areas on their own. Whatever contraceptions you have available, they'll use those contraceptives. You don't have to tell them to. They'll use them. Those women don't want to get pregnant if they have a chance for a college education and a chance to be independent and get a job and be wealthy and be entrepreneurs. That's what everybody wants. That's the automatic phenomenon when the world develops. That's what happens.
Okay, I know we've got, let's take one last question.
How does Klinefelter's fit into this?
Klinefelter's. Okay, great. So we better remind people that, so most females have 2 X chromosomes, most males have an X and a Y, and Klinefelter's is XXY. So these are men, it's about one in 1,000 men has 2 X's and a Y chromosome. And it turns out that men with, so it turns out actually, if we're asking what are the most common causes of spermatogenic failure in human populations, actually far and away, the 2 most common genetic or chromosomal causes are, one is Klinefelter's, so men who are XXY don't make sperm. And the other are the deletions on the Y chromosome. So it turns out that it turns out that males who have 2 X chromosomes, whether those males are humans or mice, don't make sperm. And we really don't understand it.
Well, but I want to add to that. We left XXY out because we could talk, it's a whole subject. We wanted to leave that. But if you want to know about XXY, that is our most favorable case for finding sperm in the testicle. Most men with XXY, and David's done all this work like with XO and Turner syndrome and Turner syndrome being the absence of a Y chromosome in what would have been a boy rather than the absence of an X in what would have been a girl. But at any rate, I can just tell you clinically that over 90% of our Klinefelter's patients have sperm in the testicle. Yeah.
Right, but it would have very few in the ejaculate.
Or nothing in the
Nothing in the
ejaculate, but it's our best prognosis. It's as good. It's a better prognosis even than a AZFC deletion.
Okay, let's thank Sherman here. Right. So Sherman, thank you for that terrific dialogue. We'll let you retreat now.
Am I supposed to go now?
Yeah. And I want to thank everybody for your great questions at this and the earlier sessions. Now my great pleasure to invite Lyurka Miller, our new, one of our BOA chair for New York, to say a few words. Lyurka. That's great.
Here I am, the person standing between you and the cocktail hour. To be taking on the role of co-chair of the BOA for Whitehead. And first and foremost, I want to thank my predecessor, Letty Gotchberg. I'm very sorry that Letty could not be with us today. She has done so much for Whitehead over so many years that she will truly be a very tough act to follow. And I am very happy to say that she will still be working with me, and we will all benefit from her energy, her knowledge, and enthusiasm, and that's really wonderful news. I also want to say to Liliana, I'm thrilled that you are my counterpart here in Boston. I really look forward to a very dynamic and productive collaboration. I would like to thank all of the faculty at Whitehead, all of the staff, particularly David Page for entrusting me with this role. And most importantly, I wish to thank all of you here for supporting Whitehead Institute. Now, today, as the day went on, several people have already told you how you can contribute to Whitehead. So you will not hear from me that you most certainly can make an individual donation. If you have, thank you, and if you're thinking about it, thank you. You will not hear from me that you might think of friends, colleagues, acquaintances that you might introduce to Whitehead because maybe they're in a position to make a donation. You absolutely will not hear from me that maybe you're aware of media outlets that perhaps could run a story and, you know, just spread a word, or you might have connections at foundations, industry, and other sources of funding. What you will hear from me is this: think about what Whitehead is giving you, what Whitehead is giving all of us. It is not just the groundbreaking research that we have heard so much about and will continue to hear about. It is not just this amazing vitality in the approach to research, this power of the curiosity and no boundaries. Whitehead is giving all of us the opportunity to learn and to grow. And it is giving us the excitement of being involved with something that's truly meaningful. So thank you for that.
Thank you, Lyrica, for all you're doing to help spread the word about Whitehead Institute. Before we depart for cocktails and other activities, I invite you to share what you've learned today with a family member, with a friend, with a colleague, with a collaborator. Your advocacy, your generous philanthropy helps to ensure Whitehead Institutes that our research programs continue. So now, if you are pre-registered for a lab tour, you'll find the details of your group assignment on the back of your name tag in blue. Now you look for blue. That is if you have pre-registered for a lab tour. So you will, if you're pre-registered and you have that blue tag, you'll find your group leader waiting for you in the lobby with a large sign showcasing the number of your lab tour. For those of you who are attending the session on our educational outreach program, please also meet your group leader in the lobby. And I invite everyone else to get a head start on cocktails in the second floor library. Thank you for joining us today. It's really been a pleasure to spend the time with you. Thank you very much.
Same thing I did.